Now, you mentioned other monoclonal antibodies, and there are some other ones out there, and I'm going to ask Dr. Zonder, what of the other monoclonal antibodies you're aware of excites you?
Well, there's two anti-CD38 antibodies, daratumumab and SAR-65984. These are, data has been presented on both of these agents, both as single agent therapy and also in combination. There were some nice oral presentations yesterday at ASH 2014 about combining these drugs with lenalidomide-based therapy. It's a similar story. These are very well tolerated medications with relatively few side effects other than infusion reactions. Particularly, you know, in the studies where they were combined with lenalidomide, it didn't seem to really change the toxicity profile at all. But particularly in a study where daratumumab was added to fairly early relapsed patients getting lenalidomide, really high response rates noted.
What do you think? Daratumumab, SAR, both good?
Yeah, I think they're both very active. They both have single-agent response rates of about 30%. Infusion reactions, again, is the most common side effect, and they both combine very nicely with lenalidomide and low-dose dexamethasone. I think it's going to be a little bit of a race to see who gets furthest quickest.
Are they the same, they have the same target, are they the same drug, or are there subtle differences between them, do you know, Dr. Kumar?
There are subtle differences. I think right now the data is based on very small studies, and there's some differences in terms of the infusion reactions, and the toxicity profile may be slightly different between the two. But I think overall both appear to be quite promising, and I think the antibodies as a whole is going to change the rules of engagement in this disease. I think it's going to be a different paradigm we're going to look at, because now we're looking at something that we could potentially combine with everything else that we have out there. So I think it'll be different.
I saw the same question asked about elotuzumab, but I assume both these monoclonal antibodies are moving into later stage testing now. Is that the case?
That is true. Especially the daratumumab is fairly advanced in terms of what they're doing with clinical trials. There's a single agent large phase 2 study that's completed accrual. There's already halfway through accrual on a phase 3 trial that's looking at the daratumumab in combination with Len-Dex. So, and they already have something planned looking at the newly diagnosed disease. So I think you're going to find broad application of this class of medications in the entire spectrum of the disease.
So let me enter a slightly more controversial area. I'm thinking again, sort of outside the United States. It sounds like we're going to have monoclonal antibodies. We'll probably have 3 of them. It's going to get kind of expensive to combine bortezomib, pomalidomide, carfilzomib, lenalidomide with an antibody, maybe with an HDAC inhibitor. How do you think we can solve that issue? Is there a solution?
I think the key is going to be, I think the antibodies, if they continue to show the efficacy that we are seeing, is going to become a partner to many combinations like what we're doing in lymphoma. So I think the key is then to find what is the least expensive combination we can find to combine with the monoclonal antibody. And I think especially combinations like bortezomib, cyclophosphamide, dexamethasone, which we know in the years to come would become much less expensive than what it is right now.
Yeah, I mean, the way I think about it is my sort of summary of lymphoma treatments over the last 10 years until the last year was anything plus rituximab is better than anything. And I suspect we're going to be that way very soon with monoclonals as well.
Very good. What about, we see a lot of excitement in solid tumor oncology and in acute lymphoblastic leukemia for immunotherapy. Dr. Berenson, are you, what's your thoughts on where we're heading with that in multiple myeloma?
I'm chuckling because that's where I started my career in Seattle, and Alex Pfeffer and Don Thomas's lab. I think we have a long way to go in T-cell therapy. I think one of the problems in myeloma is the immune deficiency. And I think we've got to deal with that before we make this effective therapy. It's different than in solid tumors.
Do you have any sense if you worked with any of the antibodies?
The antibody, you know, the checkpoint inhibitor antibodies, I think they are all just coming into the clinic. I have not yet had an opportunity to work with them. I know there has been study with the PD-1 antibody and lenalidomide, and I do not know where it stands. I think in this meeting, if you just use checkpoint inhibitor one of the checkpoint inhibitor alone, it is not that effective.
So we're probably going to have to use combinations.
Yeah,
right?
probably so.
Yeah,
but it is a bit too early for me to really say much. But we are all hoping that it would change the, it'll have a major role in myeloma and will help us especially tackle the high-risk myeloma. We are hoping an immunotherapy approach, if that is able to tackle the high-risk myeloma, that'll be fantastic.
So we've heard about oral proteasome inhibitors, particularly ixazomib and aprozomib. We didn't talk much about, but it's coming not too far behind. We've heard about 3 monoclonal antibodies, daratumumab, elotuzumab, and SAR, still waiting for a name. Just let me throw it out there a little bit. What else is it that's being developed that's exciting to you? I mean, just to get going, you talked this morning, Dr. Kumar, about a PIM kinase inhibitor. What's that, and should we be interested? What's happening?
Yeah. I think it's exciting from 2 perspectives. I think one is it's a previously unexplored pathway in this disease, and the expression of this target has been shown in myeloma cells. So I think it's a relevant target, and now we have proof of principle that a targeted medication could actually have an impact in multiple myeloma. And I think it's broadly, I think it raises the profile for targeted therapy in myeloma, especially in the context of all the new targets we are identifying from the whole genome sequencing and all the genomic approaches. So I think from that perspective, it's interesting as a proof of principle, but specifically looking at this drug, it showed some promising activity.
I'm going to throw open and just, what else do you see in the next 3 to 5 years that you want to tell people about that we haven't talked about already?
I would say,
jumped there, so
yeah, I would say intracellular JAK inhibitors, the JAK pathway. I think that's going to be heavily explored over the next year or 2. And secondly, immunoconjugates.
So the same kind of drugs we use in myelofibrosis.
Yeah, we're beginning to look at those in myeloma, but JAK1 now inhibitors, which are moving rapidly, and then immunoconjugates, both with toxins and also delivering actually immunostimulatory molecules. I think we're going to be doing a lot of work on that. We've done preclinical, but we really haven't done much in the clinic.
My bias is this, we're going to get very heavily back into immune therapy over the next few years. But I really loved what, I give Larry Boyce from Emory credit, I don't know if it was him, but to start talking about extra genomic therapy. The genome's so messed up that if we can start harnessing the immune system and ignore the genome, we're going to be a lot better off. Sundar, last thoughts?
You earlier asked about the role of genomics, personalized medicine. At this time, there are some basket trial. People have shown BRAF mutation.
Basket cases there, but you actually mean a trial called the basket trial, right?
Yeah, well, basket trial that means the patients who have that specific mutation, no matter which particular disease is their diagnosis, they are able to have access to the molecule and participate in the clinical trial. So myeloma, they have shown that BRAF mutation, you can use vemurafenib or something like that.
Thanks for bringing that up. We didn't talk about that at all. That's very exciting, isn't it? What percentage of patients have a BRAF mutation?
I think it is 5% or under, right?
46%.
And has anybody actually used a BRAF inhibitor? Does it work in myeloma?
Yes. I mean, I've not used it personally, but I've heard people who have used it have shown good responses.
Yeah. There's been one reported case which clearly shows that there's a decrease in
I actually have a patient, well, he's not my patient, but he's been to see me, but he definitely responded. And when he stopped responding, they added a MEK inhibitor, and he started responding again. Really amazing. And I think in Little Rock, they've got some data on MEK inhibitors. Okay, your turn. What's coming?
I've got 3 or 4 things, but I'll make them quick.
How about 2?
2. So the first is vaccines. I think there was some data presented here on peptide vaccines. There's also data on dendritic fusion vaccines that looks very interesting in terms of generating immune responses, and that might be the platform for PD-1 or PD-L1 to sort of augment those responses. If you think about small molecule inhibitors, BCL2 is probably a target for small subsets of patients.
And then we saw some data on that in this meeting, or at least in the scientific session.
Right.
Which specifically are you talking about here?
The ABT199 in 11-14 subsets of patients, or the more lymphoma-like subset, which again Larry has shown very nicely in his preclinical models would be a perfect setup, and we've treated a couple and seen very nice responses.
Yes, that's actually true, and some complete responses I hear too.
Yeah.
Jeff, anything?
Yeah, getting
you up in the morning?
There are, there are drugs targeting many new things. There's kinesin spindle protein inhibitors. There's selinexor, which targets nuclear pores, and both of these have been shown to have single agent activity in myeloma. And these are, I think all of us could, sort of list off our 2 or 3 favorites, but that we've worked with.
Well, I'm glad I asked because RAF inhibitors, ABT199 for BCL2 overexpressing, and selinexor. Actually, I was at a talk this morning where that was actually the most active agent in a myeloma mouse model, which is quite predictive for clinical success. So, well, I think we'll close there. I think what you can get the sense of is there's really quite a lot of excitement around what's coming. It's been a great discussion. We've reviewed and we've discussed a lot of information on the treatment of multiple myeloma. So I think for now we'll close. I'd like to thank all of you for your time and your energy this afternoon. So thank you very much.